So now it's time for Weng and Peng from the INL lab. He will open this session in nanobiosensing for personalized medicine. He's going to talk about NMR techniques for basic technologies for biomedicine.
One and a half years ago. So I've just changed the title a bit so as not to collide into a colleague who is doing next generations in sequencing, which give a bit of connotations about genetics. So I'm going to. Some of you might have seen the slides before, so just bear with me. So I'm going to have a couple of new slides at the end of these talks. But I would like to share this, why am I here and why am I doing all these things and not just a couple of work focusing. So I guess a lot of us will agree that two of the most challenging and scientific breakthrough in a common history time is actually the Moore's laws, and the other ones will be the Human Genome Projects. If you put the two things together into perspective, you will see that the price of each is falling down. And that prompts you to think about what is next. And this is what happened when I was in Beijing. By the way, I'm not Chinese or I'm not related to Huawei. When I was in Beijing, and it happens to have a sandstorm, it's common to actually experience that. And that prompts me to think that if you have the same genetic pools and you have these environments that plays a lot of either macro environments or the micro environment, which play a lot of roles that can screw out the genes that you have. And so you might have some susceptibility towards certain genes. And we might as well be able to phenotype ourselves rather than to look at just the genes, because at the end of the day you will have to put the gene into an environment and not into isolations. That's what a lot of study has shown. And you have one hand which is biology driven. There's a lot of omics here, which contains a vast information of database. So I have the privilege to come from physics department, so my talk is going to be a bit weird. So I involved in developing devices. So on the other hand, you have technology-driven personalized medicines. So you put that together. So you are sort of putting omics and onics into convenient marriage. So these are a couple of things that I have done, including miniaturizing the NMR, and that's the start of the journey. That has helped me to look beyond on just instrumentations. But I'm not going to talk about that in details because I think the organizer just gave me 15 minutes. So blame it to the organizer. And a lot of people knows about NMR as a big machine. But the question is that has been burning in me when I was a student is it possible for us to just use the physics of magnetic resonance to make it small? And that the engineering doesn't say that there's no way to make it smaller. And this is based on the fact that physics of magnetic resonance, we can actually run very meaningful data, very meaningful experiments at the low field of NMR. If you would like to have a crash course on how to build NMR, you can email me. Basically, low field NMR started during the 1980s, and we have been put into perspective by some company which wanted to sell high field. I'm not saying that high field is not working. Well, you have one high field which is pushing through to get much higher protein resolution. But what if you don't have the money to buy high field? And can we get equally meaningful information at the time domain instead of the frequency domains. I started this project by just tackling malaria infections, which infected about 200 million new cases per year. That was the days of my postdocs, and we did this in a bunch of mouse, and we were kind of lucky because through the transitions of parasites infected in the red blood cells, which turns the hemoglobins into a diametic stage to a paramedic stage. That sort of essentially gave me free label. But my work doesn't stop there. So I continued to push this further. And that comes the day where I felt that we could make this thing run faster. And the most important thing, can we have a specific molecular fingerprints for each cells or tissues that you're measuring? And here, and at the same time, you have microfluidics which come in that to isolate the cells, which is liquid biopsies, which come into popularity. So you have one hand, microfluidics which helps you to do the sample preparations, and we will do the readout. And here's the measurements of T1, T2 measurement. You can imagine this as the XYZ, but this is in the frame of T1 and T2, which is locked inside the magnetic frames. So imagine that you are able to change the glass, and if you change the glass that you are able to see magnetic susceptibility, then this is what you will be seeing. I work in, this is the results that's from the wild type. And what I can control is that I use a bunch of chemical and push the stage of the hemoglobin from the low stage to high stage of magnetic fields. And essentially what you can see here, the deoxyhemoglobin is what you get from the fMRI. This result is not published yet. And I was challenged that, could you see any differences if we will be given you a bunch of pathological state cells? And that happens to be a bunch of genetic mutated cells, which the doctors know as thalassaemic cells. So I was given the 3 different HB variants, HBD variants, and the HB variants. And you can see that all this has very specific fingerprinting. What is more important thing is that not only we can differentiate between the pathological states and the physiological states, we want to know if the 2 different persons who is not infected with, who are healthy, equally healthy, would they have a specific fingerprint? And the short answer is yes, they do have a very specific fingerprint. And this story get a bit more complicated because as you submit to good ranking journals, and you get a bit more frustrated because you're forced to give a lot more proof. And now here you have a bunch of data which you are not able to just do the student t-test and things like that. How do you differentiate between a very minor change in the fingerprint from my left-hand side or my right-hand side? So there's no clear answer, right? So that happens a day when I bump into this AI, and I know that the AI is the solutions. If we have the machines to help us to generate the results, we use back the machines to help us to interpret the results. And here's the journey of the AI. As you can see clearly, we're using a classifier called multi-dimensional scalings. What I essentially do is that I transform from a 3D counterplot of a 2D NMR back into 2D. Our brain is only able to interpret data in 2D. That's very bad. But we could have one day n scales of multiple marker. And how do we assist these doctors to make decisions? And AI could be one solution. The good thing about AI is that it doesn't only tell you quantitatively how different they are from the different subsets, but they also tell you how far or how near you are from your coffin, literally. All right, and here are 4 different classifiers that I use for my testing results, and I compared it to the performance of 5 human beings. These are some of my colleagues here. Let's actually choose the 4 best results compared to a bunch, because I simply do not want to see that humans was losing so much. And the last time when I see a human lost to a machine, it's the Deep Blue. That was about 15 years back, 20 years back, when I was a kid. And that's the last slide. Now you have a technology which runs at a POCT, point of care, that can run very fast, in minutes. So our goal is to make it real time in future. And then that helps you. The good things about POCT is that it helps you to do some form of monitoring diseases. And if you borrow the idea of AI, that can help you to improve the delivery of the informations. And what you do is that you have a molecular fingerprinting that I call molecular phenotypes, as opposed to the clinical phenotypes. And then you will then think about the next stages. Do we need to run difficult assay, such as proteomics and genomics? And welcome to the personalized medicines. Once you have the ability to address the personalized medicines, you could be able to very fast to transform this same framework to population health by using the molecular fingerprinting. And here, that's the final slides. Could anyone spot the difference? It's the iPhone. So engineer has done so much on the communications, but I kept asking myself, when can engineer fall into and do a lot more works on the biomedical field? Thank you very much.
Thank you very much. And so now it's time for questions. Do you have any question? The audience? No questions, don't be shy.
Don't be shy. Sarah.
Hi, Peng.
Yeah.
So I have a lot of questions, but what it comes to my mind is like, it looks like you have this platform and it's miniaturized already. What are the requirements in terms of sample amount, type of sample, how fast you process? I mean, I know from what I've seen and talked to you that it can be blood, it can be serum. Is there any amount and how fast do you process? And I mean, you have the machine. Just are you planning to really bring it to the hospital, or this stays in basic research in Vatican in 2005, or how is it going?
Well, we are talking. One of my students is actually running some malaria patients' results in ICBS. Okay. So she's streaming results. While she was giving talks, I was busy replying WhatsApp. So as you can see that this is what the technology can do right now. As in what samples can we run? You can imagine that this is actually an MRI machine. There's no limit that you can do to an MRI machine. The only thing you can do is that you get the right assay and you get the right answer. Did I answer your questions?
Yeah, sure. Thank you, Peng.
I think we have time for one more question.
Any other questions? So then, let me ask you, so what are other applications of your technique? Which other?
My next goal is to solve the cancer problem. I'm not solving, to understand the cancer problem. So I've gone to malaria. I have work published on, no, it's not published yet. It's under review right now in diabetes. So we've done this on the hemoglobin patty. So my fate is that we don't have to do solid biopsies. We could have a lot of information from the tissue biopsies, that including your white blood cells if you could be able to isolate your white blood cells and let me have the readout, I could tell you a lot of stories. Yeah, uncommon stories, not independence of omics.
Okay. Thank you very much, Peng. Let's thank again for his nice talk.